Sexual reproduction would be impossible without meiosis. Meiosis allows for the combination of DNA from two organisms to create a new daughter organism with an entirely unique genome. Unlike various forms of asexual reproduction, meiosis allows for greater genetic diversity on Earth. Meiosis occurs at a cellular level. It is amazing that such a tiny process drives change in the global biosphere. Throughout interphase, the cell grows and works. Caution though, when in interphase, the cell has not yet entered meiosis. However, it is important to understand interphase to see the cell before entering meiosis. Interphase is divided into three sections. During the G1 phase, the cell increases in mass to prepare for division. In the S phase, DNA is duplicated. During the G2 phase, the cell begins to synthesize proteins and continues to grow.
Prophase 1 is the beginning of meiosis. First, the chromatin begins to condense into distinct chromosomes. During early prophase 1, each chromosome aligns in a homologous pair, and crossing over occurs. In crossing over, the DNA molecules of a non-cystic chromatid are broken down by proteins and are rejoined to each other. Because of crossing over, each homologous pair has one or more X-shaped region called a chiasmata, where crossovers have occurred.
During metaphase 1, sister chromosomes align at the metaphase plate, a metaphorical plate in the center of the cell. Once this has occurred, the centromeres of homologous chromosomes orient themselves towards opposite ends of the cell.
Breakdown of proteins that are responsible for sister chromatid cohesion along chromatid arms allows homologs to separate. The homologs move toward opposite poles, guided by the spindle microtubules. Sister chromatid cohesion persists at the centromere, causing chromatid to move as a unit toward the same pole.
When telophase 1 begins, each half of the cell has a complete set of chromosomes. Each chromosome is composed of two sister chromatids, or one or both of the chromatids include a region of non-sister chromatid DNA. Cytokinesis is the moment where the cells split in two.
Spindle formation and movement toward the metaphase 2 plate occur during prophase 2. Unlike prophase 1, there is no condensation of chromatin or crossing over. Early in prophase 2, microtubules from one or the other side of the cell will attach the kinetochores, one at the centromere of each homologous pair. The homologous pairs will then move toward the metaphase plate.
During metaphase 2, chromosomes align in the center of the cells, along the invisible metaphase plate. Because of crossing over in meiosis 1, the two sister chromatids of each chromosome are not genetically identical. The kinetochores of sister chromatids are attached to microtubules extending from the opposite poles.
The breakdown of proteins holding the sister chromatids together at the centromere allows the chromatids to separate. The chromatids move towards opposite poles as the individual chromosomes mirroring the events of anaphase 1.
Nuclear form. The chromosomes begin decondensing into chromatin. Nuclear envelopes reappear and cytokinesis follows, splitting the cytoplasm into two.
The meiotic division of one parent cell produces 4 daughter cells, each with a haploid set of unduplicated chromosomes. The 4 daughter cells are genetically distinct from one another and from the parent cell.